Then take again a quick break and then do the final one before lunch. So the next group is Salla disease or free sialic acid storage disorders. Dr. Wasserstein is the workgroup leader. There you go.
Good morning, everybody. I'm delighted to be here to present free sialic acid storage disease. So I'll be presenting FSASD with my colleagues David Adams and Marjan Huizing from NHGRI. Here is a list of our work group, which represents people from across the United States as well as France and Finland. So the free sialic acid storage disorders are a spectrum of neurodegenerative disorders resulting from increased lysosomal storage of sialic acid. FSASD is inherited as an autosomal recessive disorder and is caused by biallelic pathogenic variants in SLC17A5. The gene product, sialin, is a transmembrane lysosomal protein whose function it is to export the charged sugar, N-acetylneuramidic acid or sialic acid, out of lysosomes. FSASD is a lysosomal storage disorder, and like all lysosomal storage disorders, there is a spectrum of clinical severity. And like many lysosomal storage disorders, we're undergoing a rebranding. So the most severe form used to be known as infantile free sialic acid storage disorder, or ISSD. We're now transitioning to the term severe FSASD, and the purpose of this transition in nomenclature is just to kind of acknowledge the allelic nature of these various phenotypes. The onset of severe FSASD is prenatal to neonatal. It's rapidly progressive, multisystemic disease with severe neurodegeneration, and death is typically at around a year of age. Clinical features can include fetal and neonatal high drops, coarse facial features, hepatosplenomegaly, cardiomegaly, failure to thrive, dysostosis multiplex, nephrotic syndrome, and again, a rapidly progressive neurodegeneration. At the other end of the spectrum is what's classically known as Salla disease, or less severe FSASD. In between is intermediate severe FSASD. And both intermediate and less severe FSASD present during infancy to early childhood. They have a more variable presentation in terms of age of onset, progression, and degree of neurovisceral involvement. We can often kind of distinguish between those with intermediate severe and less severe disease based on age of onset, rate of progression, and genotype is often very helpful here. Clinical features can include truncal ataxia, hypotonia, neurodevelopmental delay and disability, short stature, spasticity, facial coarsening, nephrotic syndrome, multiple neuroendocrine abnormalities, and brain abnormalities. So there's been a lot of momentum in the field of FSASD over the past few years, and that's largely due to the creation of the FSASD Consortium. And this began only 5 years ago, in 2017, when I saw a child in my clinic who was newly diagnosed with Salla disease, and he was diagnosed through whole exome sequencing. When I saw the child chatting with his parents, and like most parents of children with ultra-rare diseases, they were just desperate to find out more information about what to expect. And there was a really surprising lack of information available that we could share with them. So we invited the family to participate in Einstein's Rose F. Kennedy IDDRC's newly started program, which was called Operation IDD Gene Team. And Operation IDD Gene Team was created to connect families who have a poorly understood neurogenetic condition, to connect them with basic scientists from the Albert Einstein College of Medicine who are either studying that disorder or related disorders. So we invited them to participate. They were actually our first family to participate in this. And we had them meet with Steve Walkley, who you can see in that picture, and Costa Dubrenis, and they had a tutorial about what was known about FSASD. The family then called us a couple of days later and said, we're inspired. And they decided that they wanted to start a foundation called the Salla Treatment and Research Foundation, or a STAR. A Star is Born, which is really appropriate because Mom happens to be a professional opera singer. So based on this and the formation of STAR, we held our inaugural FSASD think tank a few months later. And at our think tank, we invited scientists and clinicians from 7 centers across the world to sit together and discuss what we knew about FSASD at the time, and to really brainstorm together about what the next steps would be, what we needed to do. And it was also the first opportunity for FSASD families from around the world to meet each other and to offer support. And this is a picture from that meeting. Over the next couple of years, the momentum grew, the FSASD consortium began to mature, and fundraising by STAR was critical. In 2021, we began having formal meetings. We had 8 focus group Zoom meetings. Now, instead of just 7 centers, we were up to 13 centers, and the plannings and collaborations began to take off. We started a patient registry, a knock-in mouse model was created, and an FSASD review was published. And this is a nice picture of one of those Zoom calls. And more recently, in 2022, we've had almost 10 subgroup Zoom meetings, now with 15 centers. Cellular models, including iPSCs, were created, and therapeutic targets are under investigation. And this is a picture from our hybrid full consortium meeting, which was just about 2 weeks ago. This graphic kind of represents the focus of the FSASD consortium. The consortium is now composed of 3 main groups, including STAR, the NIH, as well as academic clinical scientists from around the globe. One big focus is raising awareness for FSASD through improving diagnostic testing. Another big focus is basic and translational research to try to kind of help us understand better the pathophysiology of the FSASD disorders. Also working on identifying treatment and drug candidates, as well as natural history studies, and those studies are really kind of focusing on clinical trial readiness. This is what our consortium looks like now, representing 15 centers, 4 countries, and 40 investigators. So here's our heat map, and I think what is kind of noticeable about our heat map is that it's a lot redder than a lot of the other heat maps that we've seen today. And I think this just really reflects the youth of our organization and the youth of these organized efforts to understand FSASD. Our strengths right now are in patient advocacy and preclinical scientific development, but there's really a lot of progress that is being made in the other groups. So I'm hoping that the next time we meet, this will be a lot more in the green and blue area. And to share with you some of the challenges and progress that we've made, David Adams is first going to present the clinical aspects, and then Marjan Huizing is going to present the cellular mouse models, methods and tools, and therapeutic approaches that we've been working on. So I'll pass the buck.
Hi, I'm David Adams. I'm a pediatrician, a biochemical geneticist. I work in the NIH intramural program, and if we don't say it at another point in our talk, thank you so much for including us in this community, and to the organizers. We've learned a lot even over the last day of talks. As noted previously, the free sialic acid storage disorders are associated with the SLC17A5 gene. And the term solid disease originates from a municipality in Finland where a founder mutation is more common than in the rest of the world, and this is PR39C. It's a hypomorphic mutation that causes an intermediate to mild FSASD phenotype, And the term solid disease is frequently used for mild disease in general, even though we're, as we mentioned, we're trying to change the names a little bit. Officially, solid disease is this mutation, but it's also used for mild disease in the literature, and that's important to know. And then there's a severe form that, as with many lysosomal storage diseases, is related to gene function. This is one of, just as a biochemical geneticist, and put this in a category, this is one of a few diseases that are described for lysosomal transporters, along with cystinosis and cobalamin F. In the context of leukodystrophies, this has severe delayed myelination with a markedly thin corpus callosum, and we've heard about that in a number of other talks, so this isn't particularly specific. In the literature, there's some suggestion that that central myelination may improve over time, although it's rarely seen peripherally, even in older patients. And in the MRIs that we've reviewed for a short natural history study that I'll describe, we've essentially seen no evidence of myelination progression in the first half or 2/3 of a decade. The prevalence is interesting. There's about 250 cases worldwide. This is a slide that Dr. Huizing put together. And about 200 of them either have one or both of their alleles as R39C. We did some prevalence calculations based upon mutation frequency rates in NOMAD and came up with a rate of about 3 in a million outside of the Salla region and about 35 in a million in the Salla region. So this, what we'd really like is the incidence, but this suggests that there's quite a few people who are unidentified around the world. To support that, there are multiple ethnicities that are associated with sialic acid. We've got some patients from Brazil and other parts of the world that don't have any connection with the frequent mutation, and it's likely underdiagnosed. And to underline that, this has been added to some direct consumer testing, the R39C, and actually there have been some diagnoses made based on that direct-to-consumer testing, which is sort of an interesting way for things to pop up. In terms of diagnosis, you can measure free sialic acid in the urine. It's 10 to 100-fold above normal, depending upon the severity of the mutations. So that's available as a clinical test. And then there's a molecular test, of course, that you can do for mutations in the gene. And pathology, there are often enlarged lysosomes and fibroblasts, and there's increased fibroblast free sialic acid, but those are not really used as clinical tests very often. I'd just like to underline this probably undiagnosed, which is important because as we think about how to construct a study, should we focus just on the R39C mutation, or should we have a broader base to our investigations? The largest natural history studies have come out of the Finnish population, as you would guess. The most recent paper is the one referenced below, which builds on a 2002 paper that examined a cohort fairly soon after the disease was described. And from that most recent paper, they had 24 subjects that were aged 16 to 65. There's excess mortality after 30. It's progressive but highly variable between individuals. So we may need to look at something like a rate of progression rather than an absolute progression connected to age for that reason as an outcome measure. Motor skills overall improved until age 20 and then plateaued, which is challenging in terms of setting up a study and thinking about the time scale. Dyspraxia and dysarthria, as I demonstrate, are present in older patients, and there are marked motor deficits. And I was interested yesterday that several groups talked about motor scales that we might think about using in this population, since hypotonia and developmental delay are things that usually are present at the time of diagnosis. Usually 8 months to 2 years or so is typically when the kids get diagnosed. And ataxia is prominent in childhood, and then can be replaced by athertotic movements as the disease progresses. This is also from that paper. So the y-axes are in months. The top graph is motor development. The bottom graph is developmental age. This group used the Bailey and several other recognizable tools to make these assessments. The The x-axis is chronologic age. The blue lines are x equals y, just for my own reference. So those are added by me. But you can see that there's not a lot of progression once you get past a certain age. And interestingly, some of the patients live to be into their 60s. All of those folks essentially have one or two of the R39C mutations as opposed to the severe disease. So there's a bit of a paucity of information for pediatric patients, and given the fact that as soon as we've seen MRIs that we reviewed anyway, they have hypomyelination, it suggests that early intervention would be desirable. And so we'd like to see if collect additional longitudinal information about the young kids, and this was really touched off by the STAR organization and pulling together researchers over the last few years. So we have a pilot natural history study at the NIH Clinical Center. We do a sedated MRI, and a bunch of other associated procedures with that, laboratory testing. We collect biospecimens, have them evaluated by several subspecialists, photographs, skeletal surveys, all that sort of thing. And that data is still very preliminary. Clearly, the neurodevelopmental piece is important. And in addition, we found some unusual signals on mass spectrometry evaluation that's typically used for CDGs, and so we're following up on those to see if there's any interesting biomarkers there. It's a fairly busy week for families. You can see that their schedules are pretty blocked off. It's a bit exhausting, but we're trying to get essentially everything done in one point in time. But this is not really going to be sustainable for the longitudinal neurodevelopmental evaluation, so we have some other ideas in mind for that. One is that STAR has started work on a patient registry which would collect medical records, and we're trying to encourage them to collect DICOM MRI images as well, so that we have those available for longitudinal evaluation. And then Dr. Audrey Thurm, who's a neurodevelopmentalist also in the intramural program and has done work in autism and other cognitive impairment related disorders, has some tools that she's used where the family can either fill in a form by themselves, or that can be mentored remotely to collect that data rather than bringing families in, and we're exploring those as well. So the challenge is familiar to everybody here. The collection of the connection between disease manifestations and the cause of the disease is incompletely understood. And the primary clinical manifestations are fairly nonspecific and change slowly over time and vary from one individual to the next. And then the other decision we have to make, I alluded to earlier, is we have at present a cohort that's really dominated by one particular variant. And so should we focus our attention on that variant, or should we work on better ascertainment around the world to understand what the global population looks like? And then strengths you've heard about. Really, the consortium has injected a huge amount of energy into these efforts, and the existence of a mouse model and ongoing work on biomarker identification we'll hear more about during the next talk by Dr. Marjan Huizing. Thank you.
Okay, so then I will proceed with the challenges and successes in the FSASD basic science aspects, and we have divided this in 3 groups: cell and mouse models, methods and tools, and therapeutic approaches. So starting with the cell models, we've really started collecting human and mouse cell models for about 2 to 3 years now, and we've collected this group of about 10 human fibroblast patient cell lines, some lymphoblastoid lines, 3 iPSC lines derived from patients' fibroblasts, and which are now in the process of being transformed to neural stem cells, and we're discussing which other cell types or embryo or 3D models we should differentiate these in. Regarding mouse cell models, there are 2 mouse models available. We have embryonic fibroblast from both lines, and primary neuronal cells can be created from the mouse brain knockout at Einstein Medical Center. And we're also working on stable human cell lines with a complete knockout by CRISPR-Cas9 technology or by knockdown technology. It's still in process, and we're trying to tag endogenous SLC17A5 with CRISPR-Cas9 technology to see what this protein does in the cell. So some challenges at this point that we're working on is the in which cell types to differentiate the available iPSCs and which brain-like organoids we should work on. All of this requires specific experience. It's time-consuming. It requires personnel. Our consortium is limited in resources and personnel, so we have to make good and wise choices there. We're lucky to have included the NCAT Stem Cell Translational Lab in our consortium, which hopefully can help us advise or create some of these models. And we also have to think about creating isogenic cell lines. Now we're at the start of this differentiation process because it may be important to have a good control for the therapeutic and pathogenic or pathomechanism studies. So then regarding mouse models, a knockout mouse model has been available for quite a while and has been described in the literature. It's currently being studied at the Einstein Medical Center, and it's also available through the MRRC repository. This mouse model has a short lifespan, 1 to 3 weeks. After birth, we created at NIH with CRISPR-Cas9 technology an R39C, the Finnish mutation knock-in mouse model. This mouse model replicates the human disease. It has a longer lifespan. 5 to 7 months are actually mice way older than 7 months now. These knock-in mice show hypersialation and a thin corpus callosum, mimicking the human disease, and have increased urinary free sialic acid. Further characterization of the mouse model is ongoing. So some challenges regarding mouse models that we're facing at this time is that the knockout mice have a short lifespan, which may be difficult for therapeutic studies or for pathomechanism studies. But the knock-in mice are much more relevant for therapeutic studies. However, we need to choose wise outcome parameters in these mice, and neurobehavioral studies are being analyzed now. And we need, since we have such a small research group, limited resources, limited time, we need to make wise choices on planning using these mice for therapeutic studies. And we have to do this in a collaborative way. So then regarding methods and tools, we faced several challenges. Some of them we more or less solved. So first, access to free sialic acid assays or total sialic acid assays. We thought this was important that all consortium collaborators use the same assay, the same method, so we can compare the same sialic acid levels across different groups. So Greenwood Genetics Center, also part of our consortium, received a grant from the Star Foundation to perform free sialic acid assays at no cost for all consortium members, a great help, a great step forward. We have a small subgroup working on antibodies, recognizing human and mouse. SLC17A5, it's quite a challenge. Also some alternatives for that are being researched with this small working group, including tagging, as I said, the endogenous gene. We have to discuss biomarker discovery, again, make smart choices with our funding, resources, personnel, see which omics studies on which cell types or which tissues we need to pursue. And it would be good to characterize the stored material in lysosomes. And then we're doing a big focus on disease-specific cell assays that we could potentially use for screening drugs. So in this regard, we have already researched, we've already looked at a lot of different markers, like lysosomal membrane markers, endolysosome size, pH markers, looked at stored material, sialic acid, gangliosides, or others in lysosomes. And we're pursuing validating this already published assay where labeled alkyne or azide labeled free sialic acid is incubated with cells. Cells take them up through endocytosis, brought to lysosomes. In control cells, these labeled sialic acids get incorporated in glycans in the cells, but in FSASD cells, this labeled free sialic acid cannot exit the lysosomes because of the defective transporter and do not get incorporated. This is quite relevant this week because we detect this labeled sialic acid with the click chemistry assay. This technology just received the Nobel Prize. And then finally, therapeutic approaches. So our goal is to perform translational studies that are meaningful for patients now, for current patients. And we have identified a variety of therapeutic targets already known from other lysosomal storage disorders or other diseases. And several groups are pursuing different targets, and all in collaboration. So gene editing, especially for the R39Z finish mutation, is under investigation, or full gene therapy, substrate reduction therapy, chaperone folding therapies using 3D modeling followed by biochemical assays, the mTORC1 TFAP pathway, interacting drugs or mechanisms are being researched. We have not focused on remyelination drugs quite yet. We should get into those as well. And as I said, we're planning drug screening panels once we have a validated robust cell assay. Challenges in this is that, as in many other leukodystrophies, the exact pathomechanism remains unknown. So continued basic research is needed. Which model to use for meaningful drug testing? We testing that on cells, on organoids or mouse models. And if any of these initiatives yield a good candidate drug, how are we moving forward? Which candidate drug are we going to pursue in preclinical studies, and which models and resources are we going to use for this? So in conclusion, the chart that Melissa put up in the beginning of the meeting, we are addressing most aspects of this chart. As David said, the clinical aspects are being addressed. We're working on improved diagnosis, increased awareness. SLC17A5 is now included in many GLIA certified gene panels in the US, which is very nice, and we hope to identify more patients through that. Inclusion of the Salla FSASD in this GLIA meeting is another great success, and will increase awareness of the disease. Now, then I talked about the mouse models, the cell models, and that we're exploring therapies. So as other consortiums have shown in the past, also this FSASD consortium demonstrates again how collaborative efforts can truly accelerate therapeutic development for rare diseases. We're really only 3 years in, and we have achieved so many aspects of clinical translational research already. We have non-disclosure agreements in place, so it's freely we have free and material transfer agreements and research collaborative agreements, but we have a free exchange of data, of knowledge, and of materials, and it goes very easy. We have regular full consortium meetings, some subgroup meetings, and this really fosters high energy, committed and motivated collaborations. It's really a pleasure to have, we look forward to having these consortium meetings and telling each other what we're doing. So I think it's a way forward for rare diseases. So thank you.
Phenotyping diseases and using infantile or age-based systems is problematic in other diseases. I'm just a little bit worried about using a severity term in a rebranding. Just a little worried about using a severity term and how that might come across to patients, hearing you have severe disease or you have intermediate disease, and then whether or not that's setting up for exceptions to those types of classifications.
That's a good point. I mean, I think that the reality is that somebody with severe, with the severe phenotype, it's very obvious because it's an infantile onset, infantile fatal disorder. The concept of using, and I learned this way back when I was training, of using mild when you're talking about a disease like any kind of LSD, we think of it in the healthcare profession, yeah, it's mild, it's okay, it's milder than the others, but for the family, it's far from mild. So it's a little more sensitive way. We think it's still, it's a disease. It's not going to, you're not going to make it better by calling it one thing or the other. But I think that the less severe families still have a fatal disease on their hands. So.
I just worry about using any of those terms, mild, intermediate, or severe.
Right.
And I don't know the right answer, right? If you call it infantile, we're struggling this with other disorders too.
Absolutely, yeah.
Worry about families hearing that.
Right. And we also have, other lysosomal disorders which have type 1 or type A or whatever it is, and it's extra confusing then as well. So.
Great to see you again. I just want to remind that you don't have to have elevated sialuria. I mean, sialuria, you can have it without it, as we showed it to patients in collaborations with you. Do you check it in the spinal fluid at the NIH?
Is this one turned on? Yes. Hi, good to see you. We are routinely making sialic acid measurements in the spinal fluid. Interestingly, I brought this up with the Greenwood laboratory, which is doing testing, and they haven't had, to their knowledge, they have never had a false negative for somebody who ended up turning out to have sialic acid with the urine study, which was interesting because of that paper that described some patients that had elevated sialic acid only in the CSF. So I guess I would, on my to-do list, is to follow up with Keith Highland and Medical Neurogenetics and maybe Mayo, other places that are doing that testing, and find out if they've made some preliminary diagnoses based upon CSF and then later found out that the urine values were normal. So I think it's a good point. I don't have new information yet, but I'd like to explore that.
A136E that I saw that you are.
Yeah.
Okay. So,
Maybe I could just make a comment on both of those questions. I mean, for some diseases, an age-related terminology, late infantile, juvenile, adult, is somewhat easier for families to cope with, I think, than the terms mild and severe or A and B or 1 and 2. And then also for some other lysosomal storage diseases, as patients age, the metabolites literally disappear under certain circumstances. And so if you're measuring, say, glycosaminoglycans in an older MPS patient, you may not see them, even though the patient has the disease. And I think we all know that, but it's probably worth reinforcing it in this context.
Great, thank you. I think those are good points and something to consider as we're designing follow-up studies for older patients.
All right. Thank you all. Thanks again. Great presentations.